Hi everyone, my name is David Villar. Used to work at Iowa State University as a clinical toxicologist, and now have been working at a university in Colombia for the past 8 years. Welcome to this presentation that will focus on the cattle fever tick, which for many of you may be quite unheard of, but the principles of an integrated parasite management could well be applied to just about any type of parasite that creates problems raising livestock anywhere in the world. I chose this picture on the background of the slide to show what a healthy cattle tick laying a nice looking clutch of eggs looks like. And as you can see, those eggs have started to hatch and the larvae are starting to crawl around that petri dish. This slide summarizes everything that will be covered throughout the presentation, starting by setting the objectives of an integrated parasite management. We'll say a few things on the history of the cattle fever tick in the US and how they managed to eradicate it from the continental US. In tropical countries like Colombia, ticks play an important role maintaining endemic stability for diseases like anaplasmosis and babesiosis. So it may not be such a good idea to have cattle completely clean from ticks. And in order to control ticks, whenever drugs no longer are effective, we will briefly revise the life cycle of the tick and what other control measures can be implemented in each of those life stages. At least in Colombia, most drugs are no longer effective to control the tick. And I would like to show you the different mechanisms how the parasite has become resistant. And in fact, the control practices that farmers use have done just about everything possible to speed up that resistance problem. And finally, we will touch on the different alternatives that we can use to control the tick whenever the acaricides are no longer working. With very few exceptions, like we will show in a minute for the US, history has shown us that if we try to eradicate parasites, we are likely to fail. Parasites have coexisted with their host for thousands, if not millions of years. We can say they're part of the system and it's not really in their best interest to kill the host. So in this respect, a few parasites in the animal are usually not a problem. And the goal of an integrated parasite management should be to control or reduce them below a threshold that will not create production losses and obviously clinical disease. And to accomplish this, there is really no ABC approach like deworming every so often, which unfortunately has been the usual practice for the past decades. And my notion of an integrated parasite management, we can say, is kind of the fancy term to all those management practices that we can implement to minimize the need for using antiparasitic drugs. And I often like to use this epidemiologic triad as a model to illustrate this concept. You can prevent disease by acting on the three corners of this triangle. And this is exactly what John Snow proved in the early 19th century. He carried out what we call today as shoe leather epidemiology, going from house to house in London and counting all the deaths from cholera and determining which company was supplying water to each one of those houses. And without knowing that the cause of the death was the enterotoxic Vibrio cholera, he proved that water contaminated from the River Thames was the source of the disease. This basically tells us that we don't really need to know every detail of the pathology of the agent in order to prevent disease. And without using drugs that will kill the pathogen, we could always intervene at the level of either the host by increasing resistance with either using less susceptible individuals or breeds, by vaccinating or providing adequate nutrition. And we can also intervene at the level of the environment of the animals. In the case of John Snow, it was clear that avoiding drinking water contaminated from the River Thames was the answer. And in our case, it entails everything that we can basically do to break the free living stages of the ticks in the environment or the pastures where the cattle are grazing. They call John Snow the father of modern epidemiology for his work. which, as you can see, basically prove that you can prevent or control disease by disrupting the connection between any of these 3 points of this triangle. As I mentioned before, with very few exceptions, eradication campaigns have failed in totally eliminating parasites from any country. And one of those exceptions is the one conducted in the US that within a few decades managed to eradicate the cattle fever tick from all the southeastern states of the continental US. And without this tick, the main disease transmitted by the tick, which is babesiosis, also disappeared. And they now estimate savings of up to $3 billion a year to the cattle industry from not having this protozoal disease. And you may read more on how they make these calculations in the reference I provide here below. Here we see an old photograph from the USDA showing aspects of this cattle fever eradication campaign. Strict quarantine usually means no movement of animals in and out of infested premises. Systematic treatment of cattle with an acaricide. At the time, they use those concrete constructions called plunge dips. They kind of look like trenches on the ground, and when the animals run through this trench, they become completely immersed in the water bath with the acaricide. On the next slide, we will see a video clip of a portable dipping vat on wheels, if you like, that are now used in surveillance programs. But I would say that these concrete bunkers are kind of a relic of history now. And the third key to success of the program was Pasture Vacation, which basically refers to not introducing cattle in infested pastures until all the free-living stages of the tick have died. If you look at the map below, there is a strip of land at the border between Texas and Mexico, and they call this a permanent quarantine zone in which they keep inspectors doing regular visits of every cattle ranch to make sure that deer and cattle that swim across the Rio Grande are not reintroducing the tick from Mexico. And what happens when they find any cow infested with even one tick is shown partly on this short video clip. They will put that ranch in quarantine and run every animal through this trailer, which has a dipping vat, and they do this every 15 days for 6 months. And as you can see here, it's quite a time-consuming, for not saying expensive operation. But it's obviously going to pay off if the country is going to remain free from babesiosis. On the short video clip on the right, you can see one of the inspectors refilling up the tank with Kumafos, which is the organophosphorus pesticide they use, which, unlike the other ones, it has proved to be really stable over time if they keep the pH of that water constant. And so all they need to do is replenish the tank from one time to the next, and they stir it all up well with a power-driven air pump before they run the animal through the bath. Before addressing the mechanisms of resistance and the options available to control tick infestations, a basic understanding of the life cycle is shown on this diagram. As you can see here, it's a direct life cycle that may take any time between 2, under optimum conditions, 2 months, all the way up to 9 months if the environmental conditions are not favorable. This becomes crucial because it gives us an idea of how long you may need to vacate a pasture to clean it from the parasite. This diagram is showing the life stages on the animal, which may take between 18 to 35 days. That's from the time the tick larvae attaches to the time it falls as an engorged female. This engorged female is then spends 14 days laying up to 3,000 eggs, and after 30 days of incubation, from the time they were laid, a single larvae arises and it climbs to the tip of those grass leaves, where they can stay alive up to 2 months, give and take a few weeks depending on the weather. If the weather is cold, but not to the extent that it freezes, basically those females will either hibernate or postpone their deposition of eggs until the temperature rises again. And this obviously is going to extend the life cycle, as I mentioned earlier. So basically, you have to keep in mind that low temperatures only slow the metabolism of the parasite, but it doesn't really kill them unless it freezes. In our part of the world, we estimate that with our weather conditions, we probably have between 4 and 5 cycles a year. The way I recommend control programs for ticks depends basically on what you see on this figure. In areas of low incidence of babesiosis, like the high plains of the Andes, anywhere above 6,000 feet above over sea level, it's still too cold for the ticks to survive. And basically, if you were to bleed those cows, they're all going to be seronegative for babesia, and therefore there is no endemic stability. So the aim should be to basically eradicate and wipe out down to the last tick that you may find on any ranch. As you go down in altitude, the number of ticks is going to increase, and so is the inoculation rate of babesia. And ideally, what you want is 100% of the animals to be seropositive or seroconvert. So we achieve what we could call a state of endemic stability in which basically all the animals have been exposed, and they now have high levels of antibodies protecting them against babesia. And as you can see, when endemic stability is high, the incidence of cases is low, usually in those that have obviously not been exposed to the tick in the first year of life and will be seronegative. Obviously, the higher incidence of cases a year is going to take place in areas in between where we have many seronegative animals, and any spike in the number of ticks will be enough to inoculate enough babesia to cause disease. Basically, in areas of endemic stability or lower altitude, I usually worry when I don't see a few ticks on those cows, and I tell farmers to avoid treating animals until the numbers of ticks is high enough to cause disease by themselves. I know this is kind of a judgment call, and I tend to guide myself by the condition of the skin and the number of ticks on that animal to decide whether he should be treated or simply left alone. But you definitely want to have some ticks on those animals because to act as a vaccine for babesia, because at the moment we don't really have any vaccines available in the country. You may want to stop this slide and read it, but it basically is telling you that the role played by ticks in endemic areas is important, and the objective is to protect outbreaks of the disease by babesia because we don't really have a vaccine available. And as I say, ideally what you want is those calves being exposed to ticks prior to 9 months of age, because up until that age, and unlike other parasitic diseases, calves have an innate immunity which protects them from babesiosis. Once they pass that age, they become susceptible. If we move on to how ticks become resistant, there are several mechanisms, and here I'm going to use permethrin, which is a pyrethroid that has been well studied and for which there is probably no tick left in Colombia that can be killed anymore. Pyrethroids bind to the sodium voltage gated channel, and they open it, causing the depolarization of the parasite neurons. So let's assume that for a minute that you may need to open 50 out of 100 of those channels to kill a tick. When you start using the chemical for the first time, you will notice that not all the ticks require the same dosage. Some of them are more susceptible, and other ones require a little bit more to be killed. So the ideal dose should be one that is going to kill 100% of the population in that animal. And the reason why some of them are more susceptible than others, to make this easier to comprehend, is because there will be individuals that have 100% of those target proteins with both alleles susceptible. But as we move to the right of that curve, what happens is that we start finding that one of the alleles from one of the parents starts coding for a protein to which permethrin is unable to bind. In other words, a resistant protein. At the further end of the curve, we can still kill the parasite because you can increase the dose to bind at least 50 of those 100 target proteins. But as you can see, at this point, the resistant gene is expressed to a greater extent. Now, if you notice, there are two circles that are outside of the curve. And basically, there is a point in which we are creating so much selection pressure by repeating using that chemical over and over again that no matter how much we increase the dose, it's going to be impossible to kill those ticks because now all the target proteins have become resistant to the chemical. Obviously, those resistant parasites that are not killed by the treatment will be the ones mating and passing on their genes to the next generation that is also going to be resistant. Now, if you understood this concept, the one on this other slide is going to be kind of simple to comprehend. The question is, where does resistance emerges first? When using a drug like fenbendazole, which is very rapidly cleared from bloodstream, or when you use an insecticide like ivermectin, that as you can see, and depending on the formulation and the dosage, can stay in the body for up to 3 months. In this graph, the duration of action that kills 100% of the parasites could be expressed by the time at which the concentration in plasma is above the lethal dose, which is expressed as a blue line, and is clearly much longer for the insecticide than it is for fenbendazole. However, if we really wanted to choose one that is not going to favor the development of resistance, we'd rather have one with a curve like fenbendazole instead of one with the red line. What happens on the part of the red line, which is below the lethal dose in blue, is that we're going to kill the most susceptible worms in the population, that is, the ones with both alleles being susceptible, and the ones that are heterogeneous with one allele of each kind will survive, resulting in an overall increase of the resistant gene for the next generation. So in conclusion, if we want to an antiparasitic from the standpoint of slowing resistance, we rather choose one that acts quickly and just as fast is rapidly eliminated from the body in probably a matter of a few days. I'm also going to use this illustration to make another point, and that is how do we see resistance taking place in any one farm. This is not an overnight phenomenon, it's kind of a slow process. Let's imagine that we have killed all the double S susceptible parasites, and now we have 50% that are double R, and the other 50 is heterogeneous RS ticks. So when you apply ivermectin now, the first month in which the concentration is above the blue line, you will notice that the number of ticks killed on the animal is about 1/2 of those that should be killed. In other words, the therapeutic efficacy is reduced by half because you can only kill the heterogeneous population but not the double R ticks. And the duration of action is also reduced because in the second month that the concentrations are decreasing, now all the double S susceptibles have disappeared, and all the ticks that remain in the farm are either double R or heterogeneous, and neither of those can now be killed with those lower concentrations. And we have proved this in some of our in vivo studies in some farms, and you may read more about this in the reference provided here below. Now, to wrap up this idea and visualize what happens in any one farm, the scientific community now recognizes the importance of maintaining what's known as the population in refugia, which is basically accounts for all the parasites in a farm, including all the free-living stages that are not being exposed to an antiparasitic drug. On this illustration, which is taken from a recent manual entitled Control of Worms Sustainably, we see that when we use a drug for the first time, ideally selecting those animals that are more heavily parasitized, which may only be like 20% of the entire flock or herd. There may be a few resistant parasites, which are represented here with the red line. And at first, we will kill most of the parasites with the drug, and the next generation that will infest the animal which is represented here with a green color, will also be susceptible to the same drug. So we will still maintain efficacy. However, as we continue using the same drug, those resistant parasites, which, as I say, may be almost non-existent at the very beginning, will be the ones that are going to replicate in the animal and passing on their genes onto the next generation. And as you can deduce, over time, the number of resistant ones will tend to increase because we're removing all the susceptible ones from the farm. So the take-home message is that we should not really remove all the population in refugia, because that's going to speed up the appearance of resistance. And in fact, as we will see in a minute, the practices, at least in Colombia, of spraying the pasture with pesticides that kills all the larvae, which probably account for the largest populations of ticks in any one farm, is probably the fastest method of creating resistance in just a very short time. Because as you can imagine, you're removing the largest population of susceptible parasites in the farm all at the same time. At this point, let's say a few things about the molecular mechanisms by which tick become resistant. And the first one, and that usually confers the most resistance, is when the drug is no longer able to bind and interact with the target site. I will show an example of this type of mechanism on a diagram on the next slide, in which basically pyrethroids that bind to the voltage dependent sodium channel are no longer able to do so. And obviously this interaction that was creating an overstimulation of all the nerve impulses in the parasites are not doing anything any longer, and spraying the animal is no more effective than sprinkling mineral water, if you like. So what is another interesting thing is that there is what's called cross resistance or cross reaction between drugs that work at the same site. And we have noticed this for most pyrethroids, which is really a large family of drugs. In other words, if permethrin does not work, it's unlikely that any related pyrethroid like cypermethrin or flumethrin or deltamethrin will also work. This is obviously a major drawback in that it kind of limits the number of options that we have available to control the parasite. However, if we look at the next group of acaricides, which are the organophosphorus and carbamic compounds, even if they all work by inhibiting the enzyme acetylcholinesterase, we know that there are at least 3 different isoforms of this enzyme, at least in Rhipicephalus, and that explains why they don't really show as much cross resistance as you would expect initially. And we have shown this in some of our studies in which there is almost a complete lack of efficacy against chlorpyrifos in some farms, and those same ticks will still be very susceptible against another OP like ethion. Now, another important mechanism of resistance is detoxification, or increasing the metabolism by inducing enzymes that break down or eliminate the drug from inside the parasite. And some of the different enzymes, which, as you can see, in turn has many isoforms, are listed here. In fact, you will see that many times insecticides are combined with other compounds like piperonyl butoxide, which basically acts by blocking the activity of some of those enzymes in the parasite. And this is kind of a way to neutralize, if you like, these type of mechanisms of resistance in the parasite. And finally, the last type of mechanism is one that I don't really quite comprehend very well, but it seems to avoid the penetration of the antiparasitic drug to its site of action. This type of mechanism is somewhat similar to what happens with ivermectin in certain dog breeds like collies, that if you give them a normal dose of ivermectin, you could kill them because the protein transporters that are present in the blood-brain barrier will not recognize the drug and will allow it to penetrate all the way into the central nervous system of the dog and basically activate those GABA receptors. There are ticks that are resistant to ivermectin and that have overexpressed these transporter proteins, And obviously that's avoiding ivermectin from reaching those glutamate receptors in the tick. Now on these next two slides, we're going to see some examples of these mechanisms of resistance in ticks. And it's interesting that several of them can coexist in any one resistant population of ticks. Here we can see a diagram of the voltage-gated sodium channel, which is, as you can see, a big membrane protein, which is present in excitable cells like the neurons of the parasites and our own neurons, for that matter. These channels are responsible for the rising phase of action potentials that take place in nerve impulses. And if you notice, the protein consists of beta subunits, and there is a large alpha subunit in the center or core of the channel, which in turn has 4 domains, and each one of those domains has 6 transmembrane segments. The reason why I mention all this is because there have been 2 single mutations that have been discovered in resistant ticks against pyrethroids. One is in domain 3 and the other one is in domain 2. Both of them make the channel insensitive to pyrethroids. And not only that, but when the mutation is in domain 3, the degree of resistance is huge, requiring more than 1,000 the regular concentration of permethrin to kill that tick. Compared with any susceptible strain. However, if the mutation is found in domain 2, the degree of resistance is only 100 to 400 fold the concentration necessary to kill that tick if it were susceptible. Consequently, the phenotypic effect of each mutation differs quite a bit in terms of the degree of resistance, and you can read more about this on the reference that I'm providing here at the bottom. Obviously, when we look at the entire population of ticks in any one farm, the degree of resistance is going to depend on how many individuals are homozygous susceptible, heterozygous, that is, with alleles for both susceptible and resistant channels, and homozygous for both alleles resistant. I would like you to look at the top table to where it says the resistance ratio for 4 different strains of ticks from Mexico. The higher the resistance ratio simply means that we need to expose that strain to a concentration of whatever number you see there above the one needed to kill it if it was susceptible. And as you can see, the most resistant one would be the Corrales with the San Felipe intermediate between the Coalsacas and the Corrales. If you now look at the table below, you will notice that the most resistant strain, which is the Corrales, 97% of the individuals carry the gene mutation that confers the highest level of resistance, with only 3% being heterogeneous. For the San Felipe, which is in between the other two in terms of resistance, there are a few more heterozygous individuals, 29%, and 71 are all resistant. And if we look at the least resistant strain of the 3, which is the Coalsacas, almost all of them, 92%, carry the homozygous susceptible gene, which would indicate that the sodium channel mutation is not really the mechanism responsible for showing any degree of resistance. And in fact, if we look to the right on the bottom table, they also look at a different mechanism of resistance, which is based on the metabolism or detoxification of the permethrin. And what they found is that 100% of the individuals in this strain were carrying a gene that is overexpressed in a mutated esterase that has 5-fold greater hydrolytic activity to break down the pyrethroid. However, if you notice, the degree of resistance that this other mechanism conveys is much lower than the mutated sodium channel. So by showing you this type of studies, you can get a feeling of how complex these mechanisms of resistance can be, considering all the possible combinations that can potentially exist in the same parasite. And at least now we have these PCR-based assays that can help us detect many of these mutations in just a matter of a few days, and it can give us a pretty good idea of the degree of resistance that a given population might have. On this other slide, we see 3 methods of control for the cattle tick in Colombia. Each one of them on its own is making its contribution to speed up the problem of resistance by removing the population in refugia that maintains certain numbers of susceptible individuals in the farm. In every case here, they're applying the same type of chemical, which is fipronil. Fipronil is marketed with the trade name of Regent for use against agricultural pests. And we have the animal version that is marketed as Alliance. By applying fipronil directly on the pasture, what is happening is that the largest population of susceptible ticks in the farm, which is present as larvae in that pasture, is being wiped out at once, leaving only a few resistant ones that may always be present to be the ones that obviously be climbing to the animals and passing on their genes. As I said before, this is likely the quickest method to speed up resistance because we are kind of removing all at once the largest population of susceptible ticks on this farm. On the second case that we see here, what is happening is if you notice, they're applying alliance on the back line of that Cebu cow in that chute. And by doing so, not only are they wasting their money because this is a resistant breed to most external parasites in the tropics, but they're also treating an animal that may allow a few of those susceptible ticks to survive and therefore to maintain part of the so-called population in refugia. The fact that we call an animal resistant to ticks doesn't really mean that a few ticks will still be able to infest the animal and manage to successfully complete their cycle. We will see in a minute what's considered a resistant breed, and it doesn't really necessarily imply that it's going to repel all the ticks that jump on that animal. So an important principle to an integrated parasite management, which is applying target selective treatments to the animal that truly needs it, was obviously being ignored here. If you notice on the lower left drawing, selective treatment usually targets the young individuals that are usually the most heavily parasitized in the case of nematodes. Those are usually the ones that will benefit the most from the effect of the treatment. And at the same time, it serves to leave some untreated animals that are usually the majority in any one herd that are going to maintain a certain load of susceptible parasites. So the efficacy of the drug is not terminated so quickly. And finally, I would like you to take a look at the rural farmer, what he's doing. He's hand spraying a Regent. This is not a bad method if you cannot really afford to buy Alliance. But I would argue that the technique must ensure that you have to wet the animal thoroughly, starting with the legs and working its way all the way up around the animal, and particularly areas where the ticks concentrate, which are usually not visible in between the legs. This was not being done here. He was also distributing a 20-liter backpack sprayer more than 10 animals, when in reality each one of those backpacks is intended for only 4 or 5 animals. So in this case, the treatment dose is below the recommended one, which is another factor which is going to favor the appearance of resistance by only removing the most susceptible ones and leaving behind those that have a resistant allele. Now, for those of you that wonder what a massive infestation looks like, on this short video clip, we see a 3-year-old Simmental bull that was referred to as with repeated infestation by ticks. You can notice all the engorged ticks on that animal. This was definitely a case of, I would say, pure animal neglect. But with all the resistance to most acaricides in the area, we applied a combination of 3 different ones that work by different mechanisms. And as you can see on the short video clip below, he was living in what would look like a real paradise for any cow. In reality, this is a breed, Simmental, that doesn't really belong in tropical areas because not only of the high temperatures, but as you can see, the problems they get with these, at least with ticks. If we look at the same animal 10 days later of that severe infestation, you can notice all the skin lesions that were left by the ticks. There are many scabs throughout the body. The skin was really itchy to the point that the animal was scratching and rubbing himself against every tree and fence post on those premises, and that's where all those hemorrhagic areas come from. When we talk about the biology of ticks, we have to mention that they need suitable plants that will favor the attachment to the host. And in this case, the plants that were used as forage were the African star grass and some species of signal grass that make it really easy for that tick larvae to climb to the top and just wait for an animal to walk by and jump on it. Now, the other problem is that the wounds created by ticks become ideal grounds for fly larvae, like the screwworm, which is renowned for its living tissue properties in just about any warm-blooded animal. And in fact, this blowfly receives the name of Cochleomya hominivorax, that literally translates as man-eating blowfly. On the breast of this Brangus cow, and you can see that the lesions that were left by the tick were now breeding grounds for blowfly, and there is a clear opening in that skin by the invading screwworm larvae. Now, if we go back to the concept of applying integrated management approach, if we look at things that we could do on the part of the host, the number one by far would be to select for resistant breeds, like the cebus on that photograph, which basically have an innate or inborn or natural inherited, if you like, resistance to the tick. We call a resistant breed one that can repel 99% of the ticks that are going to climb on that animal. And a simple way to find out is to release 20,000 hungry larvae on top of that animal and basically wait to see how many will eventually manage to feed successfully and drop as engorged females. Now, considering that half of those 20,000 larvae will be males, less than 1% would equal to less than 100 larvae able to survive in that resistant gene. So if you see a cow with a hump on the shoulders, a large flap of skin below the neck, which is known as the dewlap, and they have droopy ears, you're basically looking at a pretty resistant animal against ticks. Another alternative is to select for animals within its own breed that harbor the less number of ticks. And there is really a very neat study done with a very susceptible breed called the Australian Illawarra Shorthorn, which is shown here on the lower photograph, in which they were basically culling animals with ticks and keeping those that were least infested. And within a matter of 4 generations, that resistance in the farm went from 89.2% to 99%. Basically, they achieved the same resistance as if they had crossed that animal with a pure Cebu stud bull in just 1 generation. And you can read more about these studies on the reference provided here below. Now, with regards to nutrition, there are no specific studies that relate the impact it may have in the ability of the animal to repel ticks. If we simply rely on field observations, we're always going to find a very large variation in the number of ticks within a group of animals. And my personal take on this is that the ones that have usually the poorest body condition scores seem to be the ones that tend to be most heavily infested. Another means to increase the immunity of the host is with vaccines. Believe it or not, there are vaccines against the cattle tick that not only lower the number of ticks on the animal, but also lowers their reproductive capacity. In other words, those ticks that still manage to drop as engorged females are going to produce less number of eggs, and many of those are going to be infertile. Here you can see the Australian version, which is marketed as Tick Guard, and the Cuban one, which is known as Gavac. Unfortunately, in Colombia, the regulatory counterpart of the FDA in the US is known to grant approval and registration to many products that have no proven record or any studies of efficacy and safety. Here we see the vaccine that is currently approved for more than a decade. Not only us, but also some independent researchers outside of Colombia have shown it to be worthless. The only study available is not even presented in a scientific journal. This Acovest magazine is a local trade magazine for farmers, and they report really truly amazing results in terms of reduction in tick counts, egg production, and reduced eclosion rates. In my opinion, everything mentioned in that report is fabricated, and something smells fishy about the whole thing, but I don't think it's probably worth saying anything else about it. If we move on to another corner of the triangle, let's see what could be done to attack the parasite on the animal apart from the usual conventional drugs. I took this picture in one of the local farms after these cows were manually milked every day. Some of them would even lay down, and these chickens would simply jump on the animal to groom them from ticks. And you can notice by the color on those feathers that this one rooster was definitely well fed. And it seemed like the cow was kind of acknowledging his work and was happy to have the chicken there. There are also some wild birds in Colombia that will stay close to cattle and do the same thing. However, I would argue that using only birds is very unlikely that we're going to tip the balance to achieve a proper control of populations of ticks. And I put a question mark on those ants that are apparently carrying some eggs, because if there is really a carnivore ant or insect that would prey on the eggs of those ticks or their tiny larvae, it would no doubt be a formidable weapon that we could potentially use, at least in places where the antiparasitics are no longer working. Now, another type of biological control agents are the fungal pesticides. And there is really a large body of literature on this topic, and I would say they hold a lot of promise, but at the moment they have only really shown good results in laboratory settings, but when used on field trials, they have not really measured up to the expectations from those in vitro assays. There are a number of funguses like this Metarrhizium and Isoplia, whose spores will attach to the cuticle of the tick, and when they germinate, they penetrate and invade the structures of that tick and eventually destroy all the organs inside. The one thing about these funguses is that they're not specific to ticks, so we don't really know what their impact in other organisms that are not target and maybe beneficial might have. At least this Metarrhizium has been shown to be pathogenic to at least 200 different species of insects. Now, unfortunately, in the Colombian market, we have a large number of these so-called biological fungal pesticides that are commercially available. Some of them are approved, like the ones on the left, and the other ones are kind of in the black market without registration. In particular, the ones you see here, we have used both in a field control study, and neither one of them has showed really any efficacy whatsoever. Another one that you see on this slide, which is sold in the black market, and it can be found in just about every farm around the country. This is an injectable formulation that claims to be a natural pyrethrin against both endo and ectoparasites. We did some HPLC analysis and found a concentration of 10.5% ivermectin, and by using the dose recommended in the label, basically the animals are receiving a dosage of 2,100 micrograms per kilogram body weight, which is about 4 times the maximum dose recommended by any approved ivermectin anywhere in the world. And to make things worse, the label also claims that the product doesn't create any residues on milk or the carcass of the animal, so there is no need for withdrawal times. And this is obviously a clear violation of the law, because consumers, at least in Colombia, are being exposed to this drug. In this case, ivermectin, and who knows what other ones we might be exposed. We denounced this situation several years ago to the regulatory agency, and as far as I know, nothing has been done. basically to crack down on this illicit market. Now, the other method of control, which tends to be used by resource poor farmers, not only in Colombia, and you can read those two references listed here that come from South Africa, is the application of used motor oil on the animal, the same one coming out of that vehicle. I know there is now a huge movement to recycle used motor oil to protect the environment and make sure it doesn't really end up contaminating any waterways. So I'm not really advocating its use on cattle, but it's obviously a very inexpensive method of tick control. The downside is that in order to be effective, it requires direct contact between the oil and the tick, so it can clog up the spiracles from which the tick is going to breathe. So you may need really to paint the entire animal in black if he's heavily infested. Also, another downside is that we're exposing that animal to whatever combustion products will be present, including polycyclic aromatic hydrocarbons, metals, metallic oxides, just to name a few, and many of those are strong carcinogens and obviously dangerous contaminants to the environment. What you normally see when you apply this onto the skin of the animal is obvious hair loss and dryness of that skin, which are undesirable effects. And if you use it very frequently, you're likely to cause a chronic inflammation on that skin. The picture you see here is from a Simmental bull from a local farm. He definitely had really dry, hairless, and flaky skin whenever that oil had been applied. So finally, if we move into things that could be done on the pasture to control the free living stages of ticks, we have some principles that could also be applied to many other parasites. In the case of the cattle tick, a clean or safe pasture is one that has never been grazed by cattle. This one ties up with the second one, which is to put cattle in pastures that have been grazed by other type of livestock, sheep, goats, horses, because neither one of these other species serve as a host for the cattle tick. The third one is a pasture that has not been grazed by cattle for more than 4 or 5 months. And I must point out that this is a time frame that we apply here in our tropical weather conditions. I know in the U.S. they used to vacate pastures or rest them for more than 6 months. If a pasture is heavily infested, another thing we can do to clean it is any of the following practices that are listed here, which are basically dedicating that land to anything different from grazing with animals. We could harvest that grass for making bales of hay or making a silage crop. We could renovate that pasture using tillage practices. It seems logical that any mechanical agitation of the soil will bury and kill the free living stages of most parasites. We could also rotate the use of a pasture with planting row crops. And finally, there are some options like burning and using annuals instead of perennial plants. I borrowed these two photographs from Susan Shanian from the University of Maryland with examples of two practices for the control of roundworms in small ruminants. But the idea could be equally relevant for the cattle tick. The first one is to alternate grazing between cattle and sheep, since they both don't share the same type of parasites, with very few exceptions, like the liver fluke maybe. So one species will basically serve to decontaminate the pasture from the other species' parasites. The other example is a recent row plantation of pearl millet, which is a cereal crop that stands drought conditions better than other ones like wheat or corn. And obviously, when you establish a pasture from scratch by planting the seed, it can be considered free from parasites. One option that was not included on the previous list was using alternative forages. This has been proved to reduce the parasite load of roundworms in sheep and goats. but unfortunately, we don't really have similar good experiences to control the cattle tick. Just as I mentioned with the fungal pesticides, there are really good studies showing good results in vitro, but they were all done in laboratory settings with experimental plots. But when taken to field conditions, they didn't really perform as expected. One of these studies was published in Nature back in 1982. They evaluated the anti-tick properties of different species of the tropical alfalfa Stylothanthis, and they did so by infesting experimental plots with a known number of larvae, and then they dragged a flannel cloth to see how many of those ticks could be recovered from the top of the plants. The stems and leaves on these plants are basically covered with what are called glandular trichomes that produce sticky secretions, and those secretions are either lethal to the larvae or they simply trap them, preventing from ascending further up. These are obviously mechanisms of defense that the plant has against feeding insects, and I would say that the effect on ticks is probably should be considered incidental. My opinion is that in order for some of these plants to have a potential use in field conditions, they would probably need to dominate among any of the other grass species on any pasture, so that you don't leave ticks with any other options but to try to climb on these ones. Otherwise, the impact will probably be limited. But having said this, some of these plants, like the Stylothanthis, they are excellent tropical pastures. They're native to South America, and they have really high nutritional value for cattle. So if a large percentage of the ground was covered on any field, I could say that this is definitely a good way for any integrated parasite management for the tick. There are also some grass species like this gamba grass with some papers published. Again, significant effect in experimental plots, but not so much on field conditions. And finally, there is another grass species. This one is native to Africa, but is well established in South America, which is this molasses grass. And when this was grown as a monoculture, it was highly lethal and repellent to larvae of cattle ticks. As you can notice, it has long trichomes or hairs on the stems and leaves. Those hairs are basically going to immobilize the movements of ticks. And the other thing is this plant has a resin, which is not only responsible for the smell of the molasses, but that resin has really a strong repellent effect on ticks. Again, this grass would probably be most likely effective if it comprises a large proportion of any pasture sward. This is my final slide. I appreciate the opportunity given to the organizing committee and all the sponsors of the event. I hope you enjoyed and found the information useful to whatever your interest may be. And please feel free to contact me to the address provided here if I can be of any assistance. So thanks again, and bye-bye for now.